Gordon, can you see us?
I can see you. Can you hear me?
Yes, we can hear you, Gordon. Great to have you here. And we see your slides, so everything is set up. So whenever you're ready, just go ahead.
Okay, thank you very much, Martin. And thank you to the organizers for letting me join you virtually. I'm terribly disappointed I'm not there. So many good friends and colleagues at this fantastic conference. And unfortunately, COVID intervened and made me have to change my plans. Copenhagen is one of my favorite places in the entire world. So next time, I guess. So let me start with a disclosure that I am a co-founder of a company called Geristate Alpha. And this is a company targeting aging to develop therapeutics. And the relevance of that will be clear from what I'm going to tell you, although I am not actually showing you any data or discoveries made by this company today. So I guess historically we are a basic biology of aging lab interested in genes that determine lifespan and aging mechanisms. But it's become all too clear that what we're really doing is we're targeting aging with the hope of developing therapeutics for humans. And I personally have a bias here, and that is that if we're talking about longevity, we need to potentially start with longevity. In other words, we need to be making animals live longer in order to perhaps be on the right track to developing therapeutics that are going to do all the great things that we hope they're going to do. So if you're targeting longevity itself, we're talking about lifespan. And if we think about the various organisms that we use in the laboratory, it's quite limited as to what you can do. And really the only organism that you can do sort of high throughput screens or mid throughput screens on longevity itself, on lifespan, is our friend, the Ceremonial species and strains. And it seems like a long way to go from here to thinking about clinical trials, as we heard about yesterday. I'm going to argue that actually it's a really good starting point, and it's not inevitable that we'll find drugs for aging and age-related disease here, but I actually think it's very likely. So this is a simple animal, millimeter in size, less than 1,000 cells, easy to grow in the laboratory. And the great thing, of course, is that we know about C. elegans that lifespan is highly plastic. We've been able to manipulate lifespan in this organism over the last 25 years. We know of hundreds of protein targets already. So we could already go out there and think about the kind of compounds that might be engaging in aging processes. We also know that many of the processes that have been discovered in the worm are likely to be also active in mammals, including ourselves. And as an aside, we also know, going back 20 years, that we can actually use drug-like molecules to extend lifespan as well. So it's not just genetics, it's small molecules and chemicals that resemble drugs that can do this trick. So we've been, as I said, particularly interested in lifespan enhancement as a sort of as a starting point, that maybe you're on the right route. If you can extend the lifespan of an animal, chances are you're doing something about a basic aging process. The worm's also been used for various age-related disease pathologies as well. You know, Alzheimer's, Parkinson's, diabetes. You know, there's a long list of models that have been created to mimic age-related disease, and that's also a place where we can think about doing chemical screens. So we and others have looked at synthetic compounds, diverse chemical libraries. We've looked at metabolites, we've looked at nutrients, and really I think there's a very large chemical space out there that has not been explored in the biology of aging. And I think that's something we're very interested in doing, is going back to basics and asking, well, what are the kinds of chemistry that are out there that really don't have biological functions right now? And could they be, very effective in extending lifespan, preventing chronic disease? So this is the conclusion slide. If we take nothing away from this talk, this is what I'd like you to be maybe mentioning when you're having a beer later in the bar. And it's really surprising to me, but maybe less surprising as we go on and this field evolves. So the conclusion is that 40 to 50% maybe And this is based on our own experience, I should say. This is not a survey of the literature. This is based on our own experience in our lab and in both published and unpublished data. And the idea is that many compounds that induce lifespan extension in this simple, tiny little animal, Ceremonial have beneficial effects when you take them into mouse models of chronic disease. And 40 to 50% is really surprising to me because of the evolutionary distance between the nematode and mammals. And it's really surprising to me that we're that lucky, to be honest, that we can make decisions about taking compounds from screens from simple animals and seeing beneficial effects in the mouse, usually being very limited in the number of concentrations and the way that we deliver these compounds. As we just heard from Linda, it can be critical as to, when you deliver compounds and what are the long-lasting effect of these compounds. And in most of the mouse studies, you don't really get a chance to do that. You get a one-off study, usually at a single concentration. So the idea that we can move compounds from nematodes to mice with such a high hit rate, I think, is astounding. If you compare it to any other standard chemical screening protocol where you get a hit rate of something like 0.1%, this is an incredibly effective way of finding compounds that may be active in mammals. That's the take home. Now I'll show you some examples. So briefly, some published data that here we're looking at 3 compounds in different classes. Thioflavin T on the left-hand side there. This is a compound that appears in thousands of papers because it binds amyloids and is used to quantify the aggregation of amyloids such as A-beta. And that's because of a fluorescence shift that happens when it intercalates in beta sheets in A-beta and other amyloids. And this was something that was raised by Silvestre Alves in my lab, a postdoc, who thought, is there any biological function to this compound? So it's a synthetic compound. It's also related to Pittsburgh compound B, which is actually used to visualize amyloids in living humans. Second compound, alpha-ketoglutarate, well known as a TCA metabolite. It's also a cofactor of many enzymes involved in lots of different aspects of biology, so a natural metabolite. And then finally, vitamin D, which you could classify as a nutrient. And so we've been studying these in different projects over the years and following to different extents, but let me just point to some of the common features of these compounds. So this is all published data showing that Thio-T on the left here extends lifespan very effectively. The controls is the black curve, and you can see at the right concentrations, not too high, or you start to see toxicity, but at the right concentrations, the survival curves moves to the right, both mean and maximum lifespan is extended, something like 70% for the mean. AKG was shown by Hyung Lab to extend lifespan as well, and we confirmed this. It kept coming up in all sorts of chemical screens that we're doing in C. elegans as being effective as a lifespan extension compound. And then finally on the right-hand side, vitamin D. This came out of a screen that was actually a disease-based screen. It was an Alzheimer's pathology screen, and we went back and tested it for lifespan in the worm. It's quite effective. It turns out that the way that we culture worms in the lab on the agar plates, there's really no vitamin D there. And so when you bring vitamin D into the system, you see much healthier worms, and there's various aspects of how vitamin D is doing that. But this slide is just an extension of those observations. It's also lifespan. So we're also looking at increases in longevity. Looks a little complex, but if you just focus on, let's start with the left-hand graph here. So each data point is a survival analysis. So this is basically taking 20 to 50 worms on agar plates, coming into the lab each day, asking if they're alive or dead by manual observation, and plotting the survival curves you saw in the previous slide. And in this way, how we're plotting it is if you are a data point above the dotted line, then it's a lifespan extension by the percentage indicated on the axis here. So as you can see, if you look at what we call the lab strain here. So this is a strain that's been adapted to, and been grown in C. elegans labs for 60, 70 years now or more. That in this case, the AKG is very effective at extending the lifespan of that lab strain. So all the data points are above the line, essentially. And this is a collaboration with Patrick Phillips and Monica Driscoll, and it's part of the Cerebral Intervention Testing Program. You've probably heard of the mouse equivalent, the intervention testing program. And we have the same goals here. We're interested in finding compounds that are robust and reproducible amongst different labs. And in this case, the labs are color-coded on these graphs. And so AKG looks great. And one of the major aspects of our program is to look at different genetic backgrounds. And the idea here is that if you have a compound that works in multiple very different genetic backgrounds, then the chances of translating that compound into mammals and into humans may be higher. And this is a super example where we have two strains. The bottom axis here shows you strain MY16 and JU775. These are different strains of the C. elegans species. But again, you see that the lifespan increase is pretty robust and reproducible across different labs. So this is looking great for C. elegans strains. And similar data for the thioflavin T compounds. So remember, AKG is the metabolite. Thioflavin T is this synthetic compounds that binds amyloids. And we see super lifespan extension in MY16 all the way up to 100% here, and generally a very robust picture of lifespan extension. So obviously, you've just heard from Linda that the invertebrate models are fantastic for investigating mechanism, and we've done some of that for each of these compounds. I just want to give you a little slice of data here, and it's data about a common feature. And these compounds come from very different chemical screens. They're very different in their chemical nature. It's likely they engage in different chemical, protein targets, for example, but they do have some commonality. So one common feature is the extension of lifespan. A second is their ability to suppress some age-related disease pathologies. And so here's an example of a worm strain, and you're looking at kind of the midriff of the animal here. And this is a strain that's expressing human A beta, made by Chris Link at the University of Colorado many years ago now. And there are many strains like this where you express a human neurotoxic protein or other disease-related protein and see phenotypes. So this is an immunohistochemical stain. And on the left-hand side, you can see after a number of days where this worm's been cultured, expressing this protein, you essentially see aggregates. You see aggregates of these proteins. You can call them plaques if you like. And during the point at which these aggregates form, we see a remarkable phenotypic difference in the worms between these worms and controls. And that is they become paralyzed. So you can essentially look down a microscope, ask if a worm's moving or not moving, and it's essentially a readout of what this immunohistochemical stain is showing you here, which is the aggregation of this human neurotoxic peptide. So the photo on the right-hand side is worms that have been grown in the presence of the amyloid binding dye thioflavin T. And as you see, the aggregates are not forming. What you see is a general soluble green signal here of the amyloid protein. And in this case, it's in muscle. So these worms are actually moving just fine. They are not paralyzed. And so, again, you have this very useful readout of a molecular pathology of age-related disease. Now, I'm not suggesting this is a model of Alzheimer's disease, but it's certainly a model of protein aggregation of a very important protein. So here is quantification, and this slide is really aimed at showing you that, wow, despite the fact that these compounds are very different from each other, they have these common aspects: lifespan extension and now suppressing amyloid beta toxicity. So the data for thioflavin T's on the left-hand side, you can see the percentage of worms paralyzed at the point of scoring, which was probably about 48 hours into expressing the neurotoxic peptide. And you can see a dramatic suppression of that paralysis with the thioflavin T compound, consistent with what I showed you in the previous slide, that the aggregate formation is slowed down. And then we cast our eyes to the middle graph, and the same thing's happening for AKG. And then graphed in a slightly different way with different concentrations of vitamin D here, we see exactly the same thing, where we're seeing a rescue or a suppression of paralysis. So why are these compounds all having these common effects? And the reason we believe is that all three are having an effect on proteostasis. It's not just about beta amyloid. In fact, these compounds are active in various models of protein aggregation, and we suspect that it's a widespread, a proteomic effect on protein homeostasis. Indeed, with vitamin D, what we've published is that it prevents the aggregation of hundreds of proteins in the worm that become insoluble during normal aging. So this gets back to the idea that screening for lifespan extension compounds in the worm throws up interesting compounds that are maybe amenable to drug development for age-related disease. So I mentioned the 40–50% that we think is effective in mice. We'll get to that. But before that, I just want to show you this graph. And this graph is the same kind of experiment where we're looking at paralysis in a beta model. And the compounds that are listed along the bottom here are all compounds that came out of an unbiased screen for diverse synthetic compounds that extend lifespan. So all of these compounds, and there's different concentrations in here, as you can see, all of these compounds are known to extend lifespan, at least under one condition in the worm. And there's a few interesting compounds on the left-hand side that seem to increase paralysis. That's very interesting. But what's consistent with the story I'm telling you are all the compounds on the right-hand side here, which reduce the relative risk of paralysis in these models. And again, think about the hit rate here compared to any other chemical screen where you might expect to get something like 0.1% initial hits or validated hits. So we have a very, very large amplification in the hit rate by going after longevity first, by going after aging. And then you come into a disease situation like this and you see a very, very strong effect. Okay, so we have been able to move some of these compounds into mice, and I'm sure that's of much more interest to most of you. But I hope so far I've shown you that the worm is a great place to start. Just a few words on thioflavin T. We engaged in a study in collaboration with Brian Kennedy and many others at the Buck Institute. This one was led by Simon Melov. And we looked at a compound called HBX, which is a relative of thioflavin T. It's a modified compound that also binds metals. And we did a late life intervention study on a large number of animals. And critical to this study was it's longitudinal. So we're looking at the same animals, time point after time point. And in this study, we measured cardiovascular health, we measured bone health, metabolism, body weight, and so on. And briefly, the major outcome of this study was first of all a comprehensive understanding of the number of animals we really need to use to make sense of the observations of an intervention. I'd recommend you. I'll show you the paper in a second. I'd recommend you have a look at that. The practical outcome actually was an effect on bone health. So Simon was able to show that mice lose bone densities as just like humans in a very similar fashion, and he was also able to demonstrate that this compound HBX reduced the extent of bone loss. And this is the paper here, published in the Journal of Bone and Mineral Research. And one of the sentences in the abstract really strikes me, and it's that we found that HBX, which extends the lifespan in C. elegans, slowed age-related femoral bone loss in mice. And I think that's striking because if you were initiating a study to find drugs to prevent bone loss, the last place you would start is a nematode worm that doesn't have bone. And so, but here we are. We see that when we target an aging process and we find something that affects longevity, we see outputs in diverse functional domains of aging. And this is backed up by the fact that we see other effects of this compound. So it prevents aggregation, extends lifespan. prevents age-related bone loss in mice. There's a mild but significant effect on an ALS mouse model published many years ago now, and also unpublished data from Julie Anderson's lab at the Buck suggests it's protective against Alzheimer's. Compound comes from worms, is effective in bone loss, and is effective in brain disease. I think that really speaks to what this field is testing right now, is the geroscience idea that if we intervene in aging, we're going to see effects in multiple domains. Quickly, AKG. With Brian Kennedy's lab, we showed that alpha-ketoglutarate extends lifespan in mice in 2 cohorts. This is pooled female and male data here. It's a modest increase in lifespan, but what was less modest was the effect on health span. And we can really sum this up with a couple of pictures here. The mice on the right are control mice, and it's a very somewhat similar experimental design to the previous work I showed you in HBX, where we have older mice coming into the lab. We're treating them from 16 to 18 months, and then we're following their natural decline with age from there on. And in this case, we used a frailty index, and this is a multiple parameter measure of health in these animals. And you can see in the controls that the animals are, the hair is graying. They're also losing hair. They have skin conditions, and if this was a video, you would see that they're not so great moving around the cage here. They're definitely normal, older animals showing signs of aging. In contrast, the mice on the left have been fed a diet that's rich in AKG. AKG tends to decline in humans with age, and so what's probably going on here is that we're restoring AKG to perhaps normal levels, and you'll see quite startlingly the health of the animals look better, even just visually, in terms of coat color, coat quality, and movement of the animals. So this work from Azar Asadi was measured something like 31 different measures of frailty in the mice. And with Brian, we really had a close look at this data, and I think it's telling us something important and exciting. And very briefly, you can imagine in a population of mice, you have long-lived mice and short-lived mice, and you have a period of morbidity where these animals are not looking good based on the metrics that we measured. What we saw with the alpha-ketoglutarate mice was not only was lifespan extended for the population as a whole, but the period of morbidity seemed to be compressed. And that's super exciting if we're thinking about interventions in humans, that not only is this a longevity intervention, but it's also an intervention that increases health span and also compresses morbidity, which is of such a massive social and economic factor. Okay, I'm just going to end with some unpublished data, and it really shows that you don't need to start with the worm, but the worm's handy along the way. And this is work that primarily comes from Julie Anderson's lab at the Buck Institute, and has been led by Manish Chamali, a research scientist, an incredibly talented young scientist. And it was kicked off by Shankar Chinta, who's with Genentech now. And what happened here was that Julie was involved in studies on rapamycin and published a paper, I think, in Journal Neuroscience, showing that the rapamycin was activating autophagy, and this was protective in Parkinson's models. And from that, Shankar and Julie decided to do a screen of small molecules that could activate autophagy, essentially looking for a better rapamycin. And in these experiments, they fused the human transcription factor, TFEB, which is essential for regulation of autophagy and lysosomal biogenesis, to a GFP, did a simple screen of cells, human cells in vitro, and found compounds that act like this in activating TFEB transcription. They did work in mammalian cells to show that indeed target genes of TFEB were activated, and that and also went on to show that this was quite an effective starting point for drug development. So then it moved into the worm, and I'm not going to show you the data for this, and I know this is a long list of discoveries here, but much of it comes from the worm. First of all, the top line is really saying that this is a compound. It's like warfarin. It's a cumin-related structure, and it's promoting mitophagy. So we worked with Malene Hansen and showed that general bulk autophagy was not necessarily increased in flux, but there's a big effect on mitophagy. The lead compound from this extends lifespan. It's elegans, no surprising. It engages in a known pathway, a nuclear hormone pathway that is known to affect lifespan in C. elegans. It affects a whole range, suppresses a whole range of models of neurological disease in the worm. It's really the best compound we've ever seen. And we've also identified the direct target, which is a mitochondrial protein, which is exciting based on the data. This compound is now in mice and Parkinson's and AD models. And I'll close by simply showing you a couple of data slides here. Actually, let's just close. Basically, it's showing you the same things that I've been showing you, and that is that compounds that extend lifespan, for example, on the right here, are also suppressing paralysis of neurodegenerative models, and in this case, also increasing mitochondrial function. So in the interest of time, I'll leave it there and just suggest that despite the evolutionary distance of this animal to humans, it's providing a series of compelling structures that we think are the basis for drug development. Our funders, and I think I mentioned all the people involved in the work as we went along. Thank you very much.
Thank you. Thank you. Thank you, Gordon. Thank you for a fantastic talk, Gordon. We are over time, unfortunately, but I really encourage people to ask questions to Gordon on Slack, and then you can answer. Gordon, I know it's very late where you're at, and sincerely appreciate your fantastic talk, but we have to move on. We are on a tight schedule. So thank you so much. Let's give Gordon another round of applause.
Thank you.
Thank you so much.